Good morning or afternoon. I'm Michael Gillings. I'm a Professor of Molecular Evolution at Macquarie University, and my main research interest is in human impacts on the living world. How does what humans do affect all of the other organisms on the planet? What I'm going to talk about today is microbial dark matter. And by this I mean the unsuspected world of microorganisms that we're only just beginning to explore. As biologists, we usually concentrate on large organisms, and because microorganisms are not readily observable, we have a bit of difficulty discovering what they do and how they do it. So I'm just going to run you very quickly through what we know about the microbial world and what we've discovered recently. So, what is dark matter? The first question is, what is out there? And we know that all of the organisms that scientists have captured in the laboratory only represent about 1% of the organisms that are in the environment. So if we go to soil or water or plant surfaces or anywhere else, only about 1 in 100 of the cells in those environments can we actually grow in the laboratory and look at, characterize. In 1987, we knew about 12 different bacterial phyla. That's large groups of organisms that are related. Just 12. Since then, what we've started to do is use DNA to investigate the diversity and identity of microorganisms in the environment, in soil and water and places like that. And very quickly we've expanded this number from 12 to, last year there was a paper published showing 92 different bacterial phyla, including a whole series of organisms that we have never managed to capture in the laboratory, and organisms that can only be found in the environment, and we can only study them in the environment. So this entire branch of the tree here is all unculturable organisms. And by the way, you and I are down here on the tips of this tree, along with all the plants and all the fungi. Okay, so let's put this in context. There's 92 phyla of bacteria, but there's only 4 phyla of plants, mosses through to flowering plants, and there's only about 6 major phyla of invertebrates and 30 animal phyla in total. So clearly the bacterial world contains an enormous diversity of organisms, 3 times the total number of phyla that we find in the animal kingdom. And animals and plants and fungi are a tiny fraction of the diversity of life on the planet. So here's a tree of all of the organisms on the planet, and mushrooms, humans, and corn plants, maize, are all just here. A tiny little sub-branch on this tree. All the rest of this is microorganisms. So that shows you our place in the diversity of life on the planet. Animals and plants are just a tiny amount of the diversity, and most of the diversity on the planet is actually microbial. So the second part of dark matter is, where are these things? Where are these organisms? And the answer is, they're everywhere, and they're in places that we would have never suspected they would be. And the reason for that is that microorganisms can tolerate wide ranges of environmental conditions, and they're also extremely versatile. So for instance, microorganisms live in very salty lakes that are highly alkaline, so high pH environments. They also live in boiling volcanic springs. Or they live in the coldest places on Earth, at the North and South Pole, or encased in ice underneath completely frozen lakes. And they're growing there and living below the freezing point of water. They're also found in the very depths of the ocean. There's no light. All there is are small volcanic vents that emit sulfur, and these organisms live right next to these small volcanic vents, sometimes at temperatures of above the boiling point of water at sea level. Water doesn't boil under the pressures at the bottom of the ocean, but these organisms here grow at 122 degrees, and it's probably 3 kilometres deep in the ocean in total darkness. And they're living there, making a living by oxidising sulfur. Very unusual lifestyles. And organisms also live in the solid rock underneath our feet, down to about 3.6, 3.7 kilometres deep in rock under our feet. And in fact, these organisms that live in solid rock actually outweigh the rest of the organisms on the planet that we're much more familiar with. So they also live in water-filled caves. Here's an example that I've worked on in Australia. It's a cave under the Nullarbor Desert where there's a completely unusual microbial ecosystem living in water-filled caves, again in total darkness. Now, let's go to the third part of this exploration of dark matter, and I'm going to talk now about the dark matter of microorganisms that live inside us. So humans have about 10 to the 14 human cells. That's a 1 followed by 14 zeros. That's the number of human cells in us. But we also have roughly the same number of bacterial cells that live inside us. Mainly they live in the digestive system, but they're also present on our skin and everywhere else that's exposed to the environment. And we're only just beginning to explore these microbial cells. It turns out that they are as much a part of us as our own human cells are, and they're really important for our health and well-being. These cells have co-evolved with humans. So they've been inside us while we've been evolving into modern humanity, and things that we have done have affected the composition of the microbiota. So our activities affect what organisms live inside us. And there's a series of cultural practices, we'll call them, that are likely to affect the composition of the organisms that live inside us. The use of fire, agriculture, exposure to heavy metals, diet, particularly processed food, how we give birth and how we raise our children, sanitation and antibiotics. I'm not going to be able to touch on all of these, but I'll try and go through some of them. We can ask, what was the ancestral microbiota like? What was the original microbiota, say, 500,000 years ago? And there's a number of ways of doing that. We can compare humans with the other great apes, orangutans, chimpanzees, things like that. We can examine fossil microbiota, or we can examine the microbiota of present-day hunter-gatherers who have a diet and lifestyle more like the original humans that we have developed from. So firstly, let's look at the microbiota of the great apes. And just as you can draw a family tree of the great apes, you can also draw a family tree of the organisms inside the great apes' digestive systems. And what you find is that the human branch is actually back at 7 to 13 million years, whereas we know that humans diverged from chimps at about 5 million years. What that means is that the human microbiota has evolved faster than the microbiota of the chimpanzee or the gorilla. And that can be put down to the cultural practices that we have developed over those 5 million years. The first and possibly most important of those is the use of fire. So fire actually allows more calories to be extracted from food. If you cook food, you can get more calories from particularly plant food than you can if you eat that food raw. What that means is that you need a smaller gut to absorb the same amount of calories. If you spend less resources building a big gut, you can actually build a bigger brain, because the two most expensive tissues in any animal body are the gut and the brain. And so if you spend less on the gut because you're using fire to prepare your food, then you can grow a bigger brain. So this is how big we expect the brain and gut to be if for an animal of our size. Actually, our brain is huge and our gut is small. Of course, if you have less gut, you have less room for microbiota, and that means you have less diversity of microbiota. And that kind of explains the difference between the diversity in the great apes and humans. So this is what it looks like. This is the diversity of bacteria in wild apes, and this is the diversity of subsistence farmers in Venezuela and in Malawi. And this, down the bottom here, is the diversity in the USA. So the diversity of microbiota, the diversity of organisms in our gut, is lower in humans than it is in great apes, and it also declines with the degree of urbanization. So how big the cities and villages and etc. are that we live in. We can examine some of the later effects of human activities on the microbiota by looking at fossils, and in particular you can look at the plaque that is fossilized on human teeth. So we can take fossils and we can examine the bacteria that are in these fossils, and by doing this, a group in South Australia in the Ancient DNA Research Centre has shown that there is a significant shift in microorganisms as we move from the hunter-gatherer to Neolithic farmers and through medieval times and into modern times. And this break here corresponds with the Industrial Revolution. And that in turn corresponds with widespread increase in the availability of processed food, in particular sugar and flour. So there is a shift in the microorganisms that live in our mouths that correspond with a change in diet from farming diets to processed foods. What this means is that as we become more civilized, our gut microbiota declines in diversity. And we can show this by looking at hunter-gatherers. And the best example are the Yanomami, who were isolated for about 11,000 years. And in fact, they thought that they were the only humans on the planet. And they are still traditional hunter-gatherers, and they have a really high diversity of microorganisms in their gut. As we move to subsistence farmers, that diversity declines, and when we move to modern civilizations, that diversity declines still further. So this bottom line here is US adults. And if we look at the individual components, we find that there are groups of organisms that are entirely missing from modern humans living in urban populations. So you can see that here, each one of these columns is a particular microorganism. A black dot means that that organism is present. So each one of the horizontals is an individual. So let's take this individual here. All of these microorganisms are present in this individual's gut. There's a few missing here. But the top slice is US populations. The bottom two slices are Papua New Guinea Highlanders that have a subsistence kind of hunter-gatherer existence. The point to be made here is there's a big empty spot right here. Gut microorganisms, gut bacteria that are present in virtually every hunter-gatherer are completely missing in the US populations. Okay, so why might this have happened? Why are these organisms missing from these populations? Well, there's a number of obvious candidates, and they're listed here. The first is caesarean birth. So when we deliver babies by cesarean, we don't give them the opportunity to be naturally colonised by bacteria during natural childbirth. And the microbiota of cesarean-born babies is different from the microbiota of babies that are born vaginally. In fact, some people have now taken to inoculating cesarean babies with vaginal microbiota in an effort to change this situation. The next thing is bottle feeding. We know that formula feeding changes the microbiota to a more adult form early on in life. Instead of, if you breastfeed, you have a different set of microbiota, and that in turn changes the way that the immune system develops. Then we've got antimicrobials. So we use a lot of compounds, antibiotics and disinfectants to kill microorganisms, and all of those change the organisms that we have inside us. Every time we take antibiotics, we kill a lot of organisms, and we never recover some of those organisms. They disappear forever. Diet is another one. So the things that we eat affect what we have in our gut. That makes good sense. And we also have our own genetics. So we all differ in our genes, we all differ in how we look, that's based on our genes, but also some of those genes affect the way microbiota colonizes us. So that's still a research area that's in early development, understanding the interaction between our own DNA and the DNA of the microorganisms that live inside us. So we're just going to go through some of these quickly. The first is antibiotics. So antibiotics kill bacteria, but bacteria fight back. They do so by either mutating existing genes or stealing genes from other bacteria that then make them resistant to antibiotics. The introduction of antibiotics and the appearance of resistance often occur in very short time frames. So here we've got daptomycin introduced in 2003. By 2005 we have resistance to daptomycin. And basically every single antibiotic that was introduced, you get resistance appearing shortly afterwards. Now what that means is that our gut microbiota, at least in urbanized populations that live in cities and developed countries, we all now carry antibiotic resistance genes in our gut microbiota. And the number of those antibiotic resistance genes increases as we get older. So this is children, school children, high school children, and adults. And you can see as we become adults, we've got more and more resistance genes in our gut. Now what that means is that humans, and also our domestic animals, because we treat them with antibiotics as well, we all have this burden of antibiotic resistance genes in our gut flora. And of course all of that disappears down sewer pipes into the environment. Now, this is how this system works. When we take antibiotics, the idea is that we get to a concentration called the minimum inhibitory concentration, the MIC, that kills the organisms in our gut or that are infecting us. The antibiotics then drop off, and a lot of the antibiotics are actually excreted in urine or feces and go down through the sewer system. They're not eliminated by sewage treatment plants, and so you get this cone of antibiotic concentration stretching out into the environment. So this is a form of pollution. We pollute the environment with antibiotics. And even more importantly, we send all of those antibiotics plus the antibiotic resistance genes that are in our gut contents with all of the other pollution that humans produce, like heavy metal pollution and disinfectants, and we send those to sewage treatment plants like this. So what we have here is a giant reactor for generating new resistant organisms with new combinations of genes and new properties, because we take already existing resistant bacteria, We mix those with other resistant bacteria in a soup that consists of antibiotics and heavy metals and disinfectants and pesticides, and we put all of those into a giant pool where we're going to get enormously complex interactions between the organisms, and we're going to generate novel organisms with novel combinations of genes that have never been seen before. So this is not just a human health problem, it's a major environmental problem as well, and it's one we need to think about when we're dealing with human waste streams. So if we think about this in a global way, this problem is very much like a lot of the other problems that face humanity. It crosses national boundaries, and it's an international problem that requires international action. China is actually doing quite a lot of research in this area, and the results that are coming out of China are quite scary. So this is a map of China with the antibiotic emissions map. So this is how many antibiotics are released into the environment every year, and the red zones here are between 2,000 and 3,500 tonnes of antibiotic per year being released into the environment. Now, bear in mind that antibiotics have effects at the nanogram level. So tiny, tiny concentrations of antibiotics have big effects on biological systems. And here we're talking about 2,000 to 3,000 tonnes per year being released. So what's the result of this? Well, a clear result is that we're going to select for organisms that have antibiotic resistance genes. And we can track those antibiotic resistance genes in the environment. And we've done that by looking at the entire coastline of China. So this is 4,000 kilometres of estuaries in China. And we've looked for the presence of antibiotic resistance genes in the mud. And what we found is that there are 1 million to 100 million resistance genes per gram of mud. And that's the size of a sugar cube, about this big. So that's 1 million to 100 million resistance genes. Bear in mind, none of those genes were there 30 years ago. All of this is human activity. So, going back to the microbiota, why should we care? Why do we care that we don't have as many microorganisms in our gut as we used to? Well, the more we learn about it, the more we realise that the microbiota is an essential part of a healthy human, just as having a good liver or a good kidney is an essential part of being healthy. And more we're starting to realise that complex diseases that are difficult to explain by infection or by environment may actually be caused by changes to the populations of microorganisms that live in our gut. And there's a whole list of these. They include things like psoriasis, esophagitis, asthma, inflammatory bowel disease, even things like cardiovascular disease are now beginning to be associated with perturbations to the population of organisms that live inside our digestive system. I'm just going to go through a few of these very briefly. The first is obesity. This is a really interesting experiment with obese mice and thin mice. It turns out if you take the microbiota, the digestive microbiota out of these mice and put them into these ones, the thin mice get fat, and if you take the microbiota out of the thin mice and put them into the obese mice get skinny. Exactly the same kinds of changes in microbiota have been associated with obesity in humans. So more and more obesity in humans is being associated with changes to the organisms that are dominant in our gut. And what that does, it changes the way we deal with storing fats, it changes the way we react to insulin, which controls glucose. So obesity has a strong link with microbiota. Other difficult to treat complex syndromes like irritable bowel syndrome are also associated with alterations to gut communities. And most interestingly, and most recently, we've started to discover that the kinds of organisms that live in our gut actually affect the way we feel and behave. This work was originally done in mice where they could manipulate the gut microbiota and look at the behaviour of the mice. But there's now a strong association between the composition of the organisms in our gut and complex psychological conditions that have increased in frequency since the 1950s, since the development of, caesarean sections, bottle feeding, antibiotic, disinfectant, all of these things that affect the microbiota. There's an association with depression and anxiety, both of which have increased in frequency since the 1950s. We don't fully understand how this works yet, but we do know that there's a lot of chemical signalling between the microbiota in the gut and the brain via the vagus nerve, and also via all sorts of signalling molecules that both our own digestive system produces and the bacteria produce themselves. So there's these complex feedback loops. And we anticipate that probably over the next 10 years this is going to be a growth area in psychology and in development of pharmaceuticals, where we start to think about, well, can we purposely restore the gut microbiota and help people with their psychological problems? So you'll notice that these changes to our gut microbiota actually correspond pretty tightly with the key transition dates in the new geological period, which is called the Anthropocene. So it looks as though we're going to have a new geological period called this, it's the Anthropocene, the age of humans, and that's brought about because of the magnitude of the effects that humans have on the planet and the other organisms on the planet. The changes to our microbiota reflect the key dates of these human effects. So we start with the shift to an agricultural diet 10,000 years ago, when agriculture was first used. The Industrial Revolution corresponds to another shift in microbiota when processed food is being introduced, and also when the first selective agents for microorganisms, the first attempts to control microorganisms using mercury or arsenic were introduced. And then the final date is what's called the Great Acceleration in the 1950s, when human population started to skyrocket, but also when we started widespread use of caesarean births, bottle feeding, sanitation, antibiotics, and disinfectants. So, the human effects on microbial ecology and our gut include significant changes in our own microbiota. That also corresponds with significant changes in the distribution and abundance of different microorganisms around the world, both through our pollution events, but also through transporting microorganisms around the world. We have changed the relative roles of bacteria in the way that the planet works. We do this by using fertilizer, for instance. We change the nitrogen cycle, and in turn we then change the ability of organisms that use nitrogen or fix nitrogen to compete in the environment. We also have rapidly selected all sorts of new genes and new gene combinations by the use of antimicrobial agents and the use of disinfectants. And of course, we have a huge effect on the manipulation of biological systems around the planet, because we mould those systems towards our own uses. But anytime we try to change the way biology works, there are always unanticipated effects. And those unanticipated effects, we need to be investigating what those might be. Antibiotic use, resistance, human microbiota, all of these are complex interactions that we are yet to fully understand. And so thank you for your attention to this talk. I'm sorry I can't be there to talk to you directly. If you do need to contact me, I'm more than happy to answer questions. The contact details are here. Thank you.